All right, in this screencast, we are going to get into the first phase of cellular respiration, which is glycolysis. So glycolysis, we know what that word is. Lysis means to break. It means the splitting of sugar. So glycolysis breaks down glucose into two molecules of pyruvate. It basically breaks glucose in half. Okay, glycolysis occurs in the cytoplasm, and it does not require oxygen. It occurs whether or not oxygen is present. Okay, so it is anaerobic. Okay, and there are two major phases in glycolysis. One is the energy investment phase, where we have to put some energy in the form of ATP. And two is the energy payoff phase, where some energy is released and converted into some ATP. So let's have a look here. Let's get this guy. There we go. So here's phase one, the energy investment phase. You take a molecule of glucose, goes into glycolysis. And over the course of the first phase, we consume 2 ATP. That's why it's called energy investment. Okay. So following that phase is the energy payoff phase. Okay. 4 ATP are formed. Okay. And also some energy in the form of the intermediate NADH, which we talked about in the previous screencast, carries some high energy electrons to the electron transport chain. So NADH is produced, and our end product, glucose, is changed into 2 pyruvate molecules. And so we have one glucose molecule at the end, these 2 pyruvate molecules. And those 2 pyruvate molecules go to the mitochondria. That's not a big enough space for me to write mitochondria. They go to the mitochondria and the Krebs cycle. Krebs cycle in mitochondria. Oops, that's not what I meant to do. All right, and then again, just a summary of my energy here. If we're talking about our carbohydrates, we have 1 glucose going in and 2 pyruvate coming out. So this is the in, this is the out. In form of energy, we have 4 ATP formed in my energy investment phase, but 2 ATP consumed in my, 4 formed in the energy payoff, 2 formed in the energy investment. So we have a net gain of 2 ATP. And then 2 molecules of NAD plus are reduced by some high energy electrons from glucose, and they are reduced into 2 molecules of NADH plus H plus. Okay, and I'm gonna clean up the slide a little bit and just get to where all these things are going. So I'll just clean up this stuff down here. So of our products, the pyruvate go to the mitochondria, as I said before, for the Krebs cycle. ATP are just used for cellular work. And the NADH plus H plus, I'm going to give myself a little bit more space. These guys carry their high energy electrons into the mitochondria to the electron transport chain. Okay, so now let's look at it in a little bit more detail. And we're going to look here at the beginning, the steps of glycolysis, the individual steps. And you do not need to know the individual steps, and you don't need to know what all these individual molecules are, but you do need to know kind of where these things happen, where we use up our ATP, where we form our 4 ATP, and where we form our NADH. So that's kind of what we're going to focus on. So Glycolysis, here we have glucose. Okay, and then the first step, take an enzyme called hexokinase. It's a kinase, so it transfers a phosphate onto something. It takes a phosphate from ATP and puts it onto glucose at the number 6 position to form glucose 6-phosphate. The next step takes this intermediate, glucose 6-phosphate, and an enzyme called phosphoglucoisomerase. Isomerase makes an isomer. Again, all the enzymes just say what exactly is happening in this step. So it makes an isomer of glucose. Fructose is an isomer of glucose. So now we have fructose 6-phosphate. And what we're kind of doing in this energy investment phase, and maybe I should have said this beginning, but here we go, is we're making glucose unstable. Glucose is an incredibly stable energy storage molecule. And in order for us to be able to break it down, we have to make it more unstable. And what do? How do we make things more reactive? How do we make things unstable? We add phosphate groups. Okay, so that's what this energy investment phase is all about, is to add phosphate group to glucose to make it more unstable. Unstable/reactive. Okay, hopefully now it makes a little bit more sense. All right, so now I had fructose 6-phosphate, so now I'm here. And we're going to work on making glucose more unstable. There's phosphofructokinase. And that is, again, a kinase, so it's going to take another phosphate from ATP and add it on to fructose 6-phosphate to get fructose 1,6-bisphosphate. Bis means 2. So now there's 2 molecules of phosphate on this compound. Okay. And then I do want to talk briefly about phosphofructokinase. This, my friends, is an allosteric enzyme. Okay. We call it affectionately PFK, phosphofructokinase. So I said before, you don't need to know any of the intermediates or the enzymes, except that you need to know this one. Phosphofructokinase is a key control point for glycolysis, as allosteric enzymes usually are. Okay, so make sure that you guys know that about PFK. All right, our next step. So we take fructose 1,6-bisphosphate, and then this enzyme aldolase finally kind of clips this in half. Okay, and you can see it kind of cuts it right down the middle here. So glucose has 6 carbons, okay? And now I have 2 compounds, 2 compounds, that have 3 carbons each, okay? One is dihydroxyacetone phosphate, and the other one is glyceraldehyde 3-phosphate. Now, in this step here, this glyceraldehyde 3-phosphate, or G3P, is what we need to go on to the energy payoff phase. Okay, dihydroxyacetone, nobody cares about it. So an enzyme isomerase turns dihydroxyacetone into a second molecule of glyceraldehyde phosphate. So what goes on from this step are 2 molecules of G3P. Okay, so we started off with 1 molecule of glucose, which has 6 carbons. And now we have 2 molecules of G3P that have 3 carbons each. Okay, so that was what moves on to the energy payoff phase in step 6. So now I'm in the energy payoff phase. So now I'm going to start releasing the energy stored in glucose. I've made it kind of unstable enough so that it'll react. And I'm going to start releasing the energy stored in glucose. Okay, so what I have here is I've taken my molecule of glyceraldehyde 3-phosphate, pretend that's right here, okay, and then this enzyme triose phosphate dehydrogenase, and triose means 3 carbons in my sugar, triose. phosphate, it's a phosphorylated, has one molecule of phosphate, and then dehydrogenase. And a dehydrogenase, remember, is always going to reduce a molecule of NAD plus into a molecule of NADH. Okay, and this is my electron carrier, NAD plus, and it's going to carry high energy electrons to the electron transport chain. Okay, and in doing so, we get our molecule of the next intermediate is bisphosphoglycerate. And you'll notice that there are two of these because remember we brought two G3Ps over to this step. So that results in 2,1,3-bisphosphoglycerate. The next step, again, I have these two bisphosphoglycerates. So each one, there's a phosphoglycerol kinase, takes a phosphate molecule from this compound and transfers it on to ADP to make some ATP. This is substrate level phosphorylation because it's taking a phosphate group from a substrate and putting it on to ADP. Because again, I have two of these, I can get 2 ATP, and that gives me 2 molecules of 3-phosphoglycerate or 3PG. Okay. Next step, I think is just kind of rearranges some stuff. Okay, phosphoglycerol mutase. So we're going to rearrange some stuff here. Not anything very exciting happening there. So we'll go on to the next step. Now I have 2 phosphoglycerate, and you saw that the last step took the phosphate from here and moved it up to the 2 carbons. So now I have 2 phosphoglycerate. Over the course of generating our next intermediate, which is phosphoenolpyruvate or PEP, a couple of molecules of water are liberated. Okay, and this just turns it from again, just kind of rearranging some stuff. And then the last step is going to take the PEP, and again, I have 2 of these, and turning them into pyruvate. And over the course of that reaction, what happens is pyruvate kinase removes the phosphate group from PEP and transfers it to ADP to make 2 ATP via, again, substrate level phosphorylation. Substrate level phosphorylation. Okay. So, and you can see here, so over the course of the energy investment phase, we consumed 2 ATP. Okay. And then the energy payoff phase, we make 2 ATP here and 2 ATP here. So that's 4 ATP in the energy payoff phase. So we have a net of 2 that's a little messy there. 2 ATP. And those ATP just go and they're used for cellular work. Okay. The most important things to move on from here are those NADHs, which will go to the electron transport chain in our mitochondria. Okay. So that is it for glucose or glycolysis.